Hello friends, today we are going to discuss one of the important theme in biology, membrane structure, that means plasma membrane structure, and different membrane transport mechanisms and systems. Membrane structure and different membrane transport mechanisms and systems. Now, membranes difference to plasma membrane, which is nothing but the envelope of the cell protecting the cell from the hostile external environment. With regard to its chemical composition, broadly it consists of carbohydrates, proteins, and lipids. And with respect to lipids, say amphipathic lipids, that means both presence of both hydrophobic and hydrophilic head. Say phospholipids, glycolipids, and cholesterol with respect to animal cell membrane. In animal cell membrane, there are additional features like glycocalyx, which is nothing but a thick polysaccharide complex structure whose oligosaccharides may attach to the top the collagen of intercellular matrix. Okay, that means plasma membrane basically consists of carbohydrates, proteins, and lipids. Where lipids with respect to animal cells, say amphipathic lipids, say phospho, glyco, and cholesterol. In case of animal cell membrane, there are additional like glycocalyx. These are the basics of plasma membrane. With regard to its structure, it is one of the important topic. Membrane structure. Davison and Danieli proposed lipid bilayer model for membrane structure, that means for plasma membrane. But later onwards, the Singer and Nicholson proposed fluid mosaic model which is most recent and acceptable model for plasma membrane because it adheres to most of the standards. Okay, with regard to its structure, we will discuss point by point. Lipid bilayer means lipid compound in two layers. These are hydrophilic heads and the hydrophobic tails. These are hydrophobic tails which face each other, whereas hydrophilic heads, they face towards the exterior. This arrangement, that means hydrophobic and hydrophilic, that means the overall lipid structure is essential in different membrane transport. We'll discuss how it went. Okay, this is a lipid bilayer. This is hydrophilic head and this is hydrophobic tail. Then other component, these are intrinsic proteins and these are extrinsic proteins. Here and there the globular proteins here and there. Okay. These are intrinsic intrinsic proteins, and the extrinsic. These are globular proteins. It's a fluid mosaic structure. That means lipid bilayer proposes single Nicholson. Here, fluid with respect to lipid component, mosaic with respect to protein component. I will tell you how. You know, broadly, the proteins here are the protein component, the extrinsic and intrinsic component. So extrinsic, which are loosely held. So these are extrinsic proteins loosely held to the surface of the membrane, and they can be easily removed. For example, cytochrome C of mitochondria. Cytochrome C of mitochondria. Whereas intrinsic protein, the name it implied, they're deeply embedded. Say they're deeply embedded in the lipid bilayer, and so they cannot be easily removed except with the use of detergents and organic solvents. For example, hormone receptors, and cytochrome P45. P45. These are different, examples for extrinsic and intrinsic protein. And why it is called as mosaic? Just unlike mosaic or ceramic tail, they are not fixed. Fluid mosaic, fluid means it keep on moving, it floats. That means the membrane changes itself. Say here it's a lipid structure, it may shift to here. Flip-flop movements, they may transfer from one phase to another phase. And also, therefore, it's like a lipid ocean over which, the proteins are floating. Fluid mosaic appearance. That means they keep on changing. Simply, that means the membrane keep on changing. That's the meaning, the implied meaning of fluid mosaic, and is most standard and the acceptable form of for membrane structure. Okay, and with regard to transport mechanism, this is an important component today. So these are about basics of fluid mosaic aspects of membrane structure. With regard to transport mechanisms, membrane transport mechanisms. Plasma membrane is relatively impermeable. It acts like a barrier for different solutes, that means metabolites. So then, depending on the nature, there are three different kinds of mechanisms of transport. Say for instance, a passive transport, simply passive diffusion, then facilitated diffusion, then active transport. Passive diffusion and facilitated diffusions are completely passive. That means it doesn't involve any energy. That means ATP. This is very important. We'll discuss one by one. Okay. Passive transport. Here we have to understand what exactly the diffusion. Diffusion is movement along the concentration gradient. For example, say it's a membrane. There are 10 units with respect to one component. Here there are 4 units. This component will move from this high concentration to low concentration. This is diffusion. That means here the movement facilitated by the concentration gradient, and the direction of movement is along the concentration gradient. That means high to low, not against the concentration, which is low to high. This diffusion is the same here. Passive diffusion means here the components will come from high to low concentration. Just basics, and we will discuss with regard to structure. Okay. Then say water and gas, they move by passive diffusion. Whereas facilitated diffusion is, the name it implied, they similar to the passive diffusion. That means along the concentration gradient from one high concentration to the low concentration, but with the help of a specific carrier protein or a transport protein. For example, the transport of glucose, galactose, these are all because of a facilitate, the name it implied, facilitate. That means there should be a mediation, there should be a carrier specific protein. Facilitate diffusion. But the thing is, the common between the two is there is no involvement of energy. They move along the concentration gradient, not against. Since there is no against, there is no use of ATP. There is no utilization of ATP. Okay. Then active transport. The name it implied. The distinguishing feature between the active and the passive is it involves energy utilization because it moves against the concentration gradient. Since it is moving against the concentration gradient, it has to utilize ATP. Simply it can call active transport. Say the most important and common classical example for active transport is sodium potassium pumps. Sodium potassium pump. So it's about the basic issues of transport mechanisms. So we will discuss one by one. Passive transport, the name it implied, no energy. Simple an illustration for easy understanding. Say it's a cell membrane and there are some 10 components here and here some 2, and this component will move, that means it's called along the concentration gradient. It is called along the concentration gradient. It moves from high, that means high to low concentration gradient. This cell membrane. This is called passive transport. That means passive diffusion. That means high, there are some 10 units, and low. Then another example, say facilitated diffusion. So the name it implied, there should be a specific carrier protein or a transport protein. Concept is same. Here along the concentration gradient from high to low, no utilization of energy, but the thing is there should be a carrier, that means specific transport protein. Say for example here it is a carrier protein, say for transport of glucose or galactose, and it will reach here along the concentration gradient. Then another example, that means the active transport. The thing is against the concentration gradient. This is the substance. How? Say it has utilized ATP against the concentration gradient. This is passive diffusion, this is facilitated diffusion, and this is active transport. In case of first two, no energy. Only thing is here there will be a carrier protein. Then here there will be a utilization of energy because it is acting against the concentration gradient. This is very important. Okay. Say for example, but here we will discuss about one important mechanism, that is called ping-pong mechanism with respect to facilitated diffusion. Ping-pong, that means I told you already, in facilitated diffusion there will be a carrier protein or a transport protein which transport a substance along the concentration gradient. How it happens? And what way? That means does it transport? That means with respect to mechanism, and the most acceptable mechanism, that means proposed mechanism, is ping-pong mechanism. Ping-pong mechanism for facilitated diffusion. The most common example, glucose, galactose transport. Ping-pong means it is with respect to structural conformation of the carrier protein. That means Pong is one conformation, Ping is another conformation. Say for instance how it works. See this cell membrane. This is Pong. Later onwards it become Ping. Say how they transport. This is very important. Say there are certain say solutes. This is exterior part. This is inside the cell. Okay. This is transport protein. This is Pong form. Pong form. What exactly Pong form? The structural conformation of the transport protein is in such a way that it faces the solutes. It exposes the solutes so that the solutes come and bind here. That means just like face the solute so that solutes come and bind here. Okay. Then when it binds, the Pong become Ping. That means it's become here by structural conformation. That means it simply transports the solutes to inside the cell. Low concentration. This is high concentration. The Ping form exposes to the low concentration phase. That means where it delivers, where it transports the subsolutes. Transport the solutes facing here. That means low concentration. Whereas Pong facing the high concentration. That's it. This ping pong mechanism is the most acceptable basis for facilitated diffusion. For example, you know how it works? Insulin increases, that means it is under control of hormones. That means facilitated diffusion is under the control of hormones. For example, insulin increases glucose transport in muscle adipose tissue. Then amino acids in case of liver and other tissues. Here insulin increasing the transport, that means and stimulating the facilitated diffusion. Okay, it's about ping pong mechanism of facilitated diffusion. And we have discussed at length about active transport because it is most important component. Active transport is most important component for the survival of our life, for the survival of the cell. How? For example, say, I told you already, active transport is against the concentration gradient. And third form of mechanism, the most common example is sodium potassium pump. Here pump is with respect to ATPase. Say for instance, this is outside and this is inside. Lies ATP, I told you already. It is inside of the cell, it is external. Here higher K plus concentration, whereas outside higher sodium concentration, higher K plus. That means simply how it reacts. Say 3 sodium outside plus inside plus 2 potassium outside plus ATP gives rise to 3 sodium outside plus 2 potassium inside plus ADP plus PA. It's a simple representation of sodium potassium pump. We have to discuss at length about this point, you know. I told you already, sodium potassium pump, which is a primary active transport. Nothing but primary active transport. Why? It is essential for the survival of the cell. You know, in our cells, there should be high intracellular potassium concentration. High intracellular potassium concentration. Why? Why we require high K plus? Because K plus ion is essential for pyruvate kinase. Pyruvate kinase, which is required for glycolysis, required for protein synthesis, and most importantly, sodium potassium concentration gradient is required for nerve impulse conduction, simply called action potential. You know, pyruvate kinase is one of the important enzymes in glycolysis that in the long run leads to ATP production. For that, K plus is required, and for protein synthesis, K plus is required. And most important, we have discussed in the last class, that means histone physiology, regarding nerve impulse conduction, what we call action potential. The concentration gradient, that means outside the cell there should be positive. Say this is nerve, this is neuron. Outside it should be positive, and inside it should be negative. That means minus 70 millivolts. That means more number of sodium ions has to be expelled out. Then it will lead to action potential. It will jump. You know, it's about saltatory conduction. We have discussed already about this concept. That means in order to facilitate the action potential, that means nerve impulse conduction, there should be concentration gradient of sodium and potassium across the neuronal membrane. That means less. That means negative inside, positive outside. That means positive has to be expelled, or that means sodium out of the cell. That means with respect to axon, I am saying neuronal inside exoplasm. This is important. Without this arrangement, without this concentration gradient of sodium and potassium, you are not going to expect. We are not going to expect the action potential. Without the potassium, there is no pyruvate kinase. Without pyruvate kinase, there is no glycolysis. Without glycolysis, there is no ATP. Without potassium, there is no protein. That means the cell shall start to die. That is why I told you, potassium, higher intracellular potassium concentration is essential. For the survival of life. That means, who will facilitate that? Sodium potassium pump. How? I told you, na? This sodium potassium pump, which is ATP driven, it will remove 3 sodium from inside of the cell to outside, and will intake 2 potassium from outside of the cell to inside. It's all a technical example for co-transport, you know. 3 sodium from inside of the cell removed to the outside, whereas outside it will take 2 potassium and will take 2 inside. That is how it build up higher intracellular potassium concentration, and is how it will remove the sodium concentration. That is how it removes the concentration gradient. That is how it maintains the essentiality of the life. It is very important. That means it is like sodium potassium pump is a crux point of life. We can conclude such an inference. Okay, and it is this is how the simple reaction: 3 sodium inside plus 2 potassium outside plus ATP, plus 3 sodium outside plus 2 potassium inside plus ATP plus PA. This is a basic understanding about sodium potassium pump. You know, there is one more important thing. Our brain. Our brain is steroid derivative extracting from seeds of African shrubs. This will inhibit sodium potassium pump. It's very important. Our brain is a steroid extracting from the seeds of African shrub. You know, the hunters in Africa, they will tip the arrows, say it is arrow, they will tip the arrows with our bend so that they may use it to hunt the animals. I told you already, without sodium potassium pump there is no life, there is no existence of cell life. Since there is no existence of cell life, they use the same to hunt the animals. This is very important. This is very important in a steroidal component. Then regarding other systems, what we call transport systems. It's all about mechanisms, and now we have discussed about transport systems. Membrane transport systems. There are broadly of three types again. Uniport, symport, antiport. For illustration, the cell membrane, this is uniport, this is one component, a single molecule. Then this symport, this is one, and this is another. Antiport is in one direction, and this will be in opposite direction. This is uniport, this is symport, and this is antiport. Say glucose transport examples, in erythrocytes. Then this is most important, sodium and glucose transport into intestinal mucosal cells from gut. With respect to antiport, exchange of chloride and bicarbonate ions in erythrocytes. That means chloride shift. These are all the secondary active transport mechanism. Active transport systems, okay? Uniport, the name it implied, one single molecule. The single molecule will transport from one end to another end. That's it. Say the glucose transport in erythrocytes. Generally, there will be a regular transport of glucose into the erythrocyte. This is simply the uniport system of transport in a membrane. That means uniport membrane transport system. Then symport. The word it is sim, two. That means simultaneous transport of two different molecules or substances in one direction. This is very important. In one direction. In one direction. That is very important. That means this is one substance, this is one different molecule, this is another one. They are both transporting in one direction. Say sodium glucose transport, they transport into the interstitial mucal cells from the gut, both in one direction. Sodium and glucose in one direction, one direction. Then antiport, the name it implies, anti, in opposite direction. The reversal of symport, in opposite direction. That means it is one different molecule, it is another different molecule. One come in one direction, then anti-direction. That means against or opposite direction. This is 1 and 2. The antiport. Say that means this is a popular phenomenon called chloride shift. Exchange of chloride ions and bicarbonate ions in erythrocytes is through an antiport. That means, so these are all the secondary active transport systems, wherein symport and anti-transport together considered as co-transport systems. This is very important. If one asks you co-transport, you have to ask the second question, whether it is symport or antiport. Antiport means whether it is both two molecules moving in one direction or the two molecules moving in opposite direction. If it is opposite direction, it is antiport. If it is the same direction, it is symport. Okay, this is about different systems. And last important component in the present chapter is it is all related to solutes, small molecules like water. We all knew that water will transport by means of, how the water moves. That means the movement of water is just by osmosis. say from diluted solutes, let me say from low osmotic pressure to high osmotic pressure. Say how the water transport. Water is simply by passive transport only. The movement of water is simply by passive transport, what we call osmosis. Say water, they say low osmotic pressure to high osmotic pressure. Here there is no energy, this is very important. That means water moves by osmosis from low osmotic, that means dilute sample to higher, means concentrated sample, high osmotic pressure. Low osmotic pressure to high osmotic pressure. Low dilute sample to concentrated sample. Then concentrated solution, no? That's how the water moves. It's simply okay. How? It's about different things like water, solutes, metabolites, or small molecules. How about the transport of macromolecules? What's called macromolecular transport is very important. The last component, macromolecular transport. How we take, say for example, a polysaccharide, a protein, a polypeptide, say protein, a polysaccharide, a for example, polynucleotide. How they get done? There are two ways again. Endocytosis and exocytosis. That means to transport the macro or megamolecules like proteins, polysaccharides, polynucleotides, there is a different mechanism. Exocytosis, endocytosis and the exocytosis. For example, say simply endocytosis is facilitated by clathrin coated pits. Clathrin coated pits. How? For example, it is cell membrane and the desired substances. Then the next component, it forms a vesicular like structure. Then it forms ultimately and it transports inside. Is endocytosis. Then exocytosis, same, then reverse fashion. For example, say it's the LDL proteins, low density lipids. How they uptake is by this mechanism. Say if the same component, say in the form of vesicle, that means Golgi apparatus involving, what we call internal transport system of the cell is by means of vesicles. Vesicles are one of the integral component of Golgi apparatus, and these vesicles, they transport different substances whenever it is what we call secretion. The substances which are aimed to secretion is facilitated by, that means transport by vesicles, which are the integral part of Golgi apparatus. And how they will form? Simply they by means of vesicle, then they adhere, that means they will adhere to the plasma membrane, then finally they will release out. Finally they will release out. That means exocytosis is most important part in, the substance, a hormone like hormones. This is very important component. These are all important component in macromolecular transport. Simply, this is a reverse. Endo uptake plus exo. It is releasing the macromolecules outside the cell. This is how the macromolecular uptake, like protein, polysaccharides, and polynucleotides. Okay, this is about different membrane transport mechanisms and systems. This is a very important component in particular in exams point of view. You need to follow some basic reference book and a basic for consultation of the knowledge together with my presentation. Okay, you may like, comment. Thanks for watching.